Hi, I'm Nick, an MCAT instructor and content creator here at Kaplan. In this video, Ari, one of our expert instructors, is going to walk you through what we've identified as one of the toughest passages on Kaplan's free MCAT practice test. This test was designed by Kaplan's question writers and validated by our psychometrics team to be as representative as possible to the actual MCAT so that you know what you're getting into on test day. This particular passage is from the Bio-Biochem section and focuses on a specific dehydrogenase deficiency. Follow along with Ari as she shows you how to break down and attack the passage in questions using Kaplan's passage in question strategy.
Looking at passage 6, my first impression is that it's pretty short and has no figures or data. The phrase at the very beginning of the passage is one that I recognize as a familiar pattern. When an MCAT passage starts off by describing some symptoms of an individual, it's what I call a case study passage. These aren't the most common passage type, but when they do come up, they're almost always short, and the first paragraph describes some of the individual symptoms and background. The remaining paragraphs will likely go into more depth about the disease, which I can see here is probably about fat metabolism. Because I'm relatively familiar with the content of fat metabolism from my studies, I'm going to do a fairly brief analysis of the passage using Kaplan's highlighting distill method, knowing that I can head back into the passage for support with the questions. Reading through paragraph 1, I'm going to center my highlights on what I think will be relevant to fatty acid metabolism, since I know from my brief preview that's the connecting piece in paragraph 2. I'm going to highlight the phrase hypoglycemic or low blood sugar, since I know that sugar metabolism is going to be affecting how fat is metabolized. I'm also highlighting depressed urine ketone levels, since I know that when fat is broken down into acetyl-CoA, that acetyl-CoA can be converted into ketone bodies. The definition for the acronym MCAD, or medium chain acyl-CoA dehydrogenase, already stands out to my eye, so I'm not going to bother highlighting it. Paragraph 2 describes the enzyme that's dysfunctional. I'm told that MCAD is an enzyme that has a role in beta oxidation of fatty acids that range in length from 6 to 12 carbons. Since that's information I'm not familiar with, I'm going to highlight that part. The rest of the paragraph describing the ketone bodies and electron carriers is content I already know from my studies, so I'm going to skip highlighting those details. The connection between paragraph 1 and 2 shows up in the first sentence of paragraph 3. I remember the individual in paragraph 1 had low blood sugar, so I'm going to highlight this detail here about how MCAT symptoms show up under high energy demands after a blood sugar drop. The rest of the paragraph does have some more important information about how the fat accumulates and causes damage, as well as how MCAT can be screened, but I know if I highlight everything here that looks important, it will be tougher for me to spot the relevant details here for the questions, so I'll stick with just highlighting this bit and move on. I know to come back to the details in each of these paragraphs for more information if a question requires it. At first glance, question 29 has a really long question stem and no obvious patterns in the answer choices. Usually when I see a setup like this one, it's a passage-based reasoning question, and it's probably going to require some digging into the passage since I see that it's referencing MCAD. Reading the question stem, I'm given some facts about a specific patient that has MCAD. The father is a carrier, which means he carries one faulty gene, but not two. The question stem mentions that the mother is not a carrier, and I can then infer she's probably homozygous dominant, so does not have any copy of the faulty allele. So the question is asking how this genetic cross would result in a child with MCAD deficiency. First, I'm going to recall a few facts about the father being a carrier. I know that a carrier is someone who has a copy of the allele but doesn't express the phenotype. That means that MCAD must be a recessive trait. In order to get the phenotype, you need to have two faulty copies. I don't really recall from my passage analysis what would help me with this question. I don't remember anything about MCAD deficiency inheritance, so on test day, this might be a good question to skip past and do the other questions first. At this point, I'm going to do a quick scan through the passage starting with paragraph 1. This paragraph gives me the background for the case study, and the only relevant clue here is that they think the infant has MCAD deficiency because of the family history. Well, I already knew that this is a heritable trait from the question stem, so nothing new to gain here. Paragraph 2 gives me the background for what MCAT does. No real clues here. Paragraph 3 describes what happens when there is MCAT deficiency. There is a bit of a clue at the bottom where I'm told there's a genetic test for a point mutation. This gets me thinking. Since I need two faulty copies to express the disease phenotype, and I know that one copy can be coming from dad, then the other faulty copy can arise in two different ways. I can either have a point mutation in the cells that make the egg, which means that the mutation is only present in the eggs and is not present in the rest of mom's cell. Or mom passes down a normal copy of the MCAT gene, but the point mutation in MCAT occurs in the fetus super early in development. I'm not 100% sure though, so I'm going to carefully analyze the answer choices and see if something works. It ends up matching really nicely to answer choice B. Germline means the cells that are going to produce gametes, so the eggs in mom. The faulty copy of the genes would not show up in the rest of mom's cells, which is why the genetic screening in the question stem tells us that mom isn't a carrier. The other answer choices are pretty interesting because I can now see how tough this question would be if I had to read through all the answer choices. A isn't going to work because a non-disjunction means that the sister chromatids aren't separating. So yes, I'd have two copies of the faulty MCAT gene, but I'd also have two copies of a bunch of other genes as well from dad. And if you pair that up with mom, then it results in a trisomy of a bunch of genes. This first sentence in the question stem turns out to be pretty important. If the patient was otherwise healthy other than MCAT issues, then they definitely don't have a bunch of other trisomies. I can apply that same logic with C. If I have a non-disjunction from mom so that I only have one MCAT gene from dad, then it's going to create a monosomy of a bunch of other genes as well, making this person affected with more than just MCAT deficiency. The final answer choice in D is pretty close to the prediction I had about the fetus, but has an important difference. This answer choice is saying that there's multiple mutations in MCAT in somatic cells. I know from my studies that somatic means not germline, so any cell that isn't used to make gametes. I think the reasoning here is assuming that the fetus doesn't inherit any faulty copy from dad, and that both copies of the gene in the fetus get the mutation for MCAT. While I suppose that's possible, the likelihood of two separate point mutations on different alleles for the same gene at the same time is pretty low. Between answers B and D, B has one point mutation and D has two. B is a much more plausible scenario. In my initial glance through question 30, I noticed that there are graphs in the answer choices with glycogen on the x-axis and electron carriers in the legend. I know that when there are graphs as answer choices, it can be a bit overwhelming to precisely predict what's going on, so my general approach will be identifying one trend at a time, then eliminating as I go. Looking more closely at the question stem, it's asking for a person with MCAT deficiency what happens to the electron carrier levels before and after glycogen has been all used up. It's kind of a lot to break down, and there's one thing I can already spot as an area for elimination even without considering MCAT and glycogen components. I know from my studies that when NADH, the reduced form, donates its electrons, it forms NAD+, which is the oxidized form. And when FADH2 donates its electrons, it forms FAD. In other words, it will be either the oxidized or the reduced form that's high. Both cannot be high at the same time. Answer choice C has both the oxidized and reduced forms of the electron carriers high in the presence of glycogen, and both the oxidized and reduced forms low without glycogen. Even without considering the effect of glycogen, that's enough to eliminate answer choice C. All of the other answer choices have that balanced distribution, so I'll need to do a bit more digging into metabolism and the passage for more clues. The first place I'm going to go to is paragraph 2, because I recall from my passage analysis that paragraph 2 was where MCAT's role was discussed in more detail. I learned from paragraph 2 that MCAT is an enzyme that functions in beta oxidation and produces acetyl-CoA as well as the electron carriers NADH and FADH2. While I knew the products of beta oxidation already from my studies, it's nice to have that reminder here. Paragraph 3, however, has an interesting detail that I did highlight. The symptoms of MCAT deficiencies aren't always present. They are only seen when there's low blood sugar. Relating that back to the question, I know that glycogen is a stored form of glucose, and that the body prefers to use glucose for energy before anything else. So when I'm energy low and I've used up all of my glycogen stores, I would feel the effects of MCAT deficiency since I'm not able to break down fats for energy. Regardless of MCAT though, when glycogen is in excess, it means that the energy levels of the body is high. I know from my studies that molecules that signal that the energy levels are high are things like ATP, as well as the reduced form of the electron carriers NADH and FADH2. Therefore, the oxidized form, NAD plus and FAD, should be low. That allows me to eliminate answer choice B, which has that relationship flipped. Between the remaining two answers, the difference lies in after glycogen depletion, so really where MCAT deficiency comes into play. I know that if I'm out of glucose, then I have to break down fats to get my energy. If I don't have MCAT around, I can't fully break down the fats, and since I know that the products of fat breakdown include NADH and FADH2, without MCAT present, I should see low levels of NADH and FADH2 and relatively high levels of NAD plus and FAD. That matches to answer choice A. D is what I think would happen with someone who has normal MCAT. The levels of NADH and FADH2 should remain relatively the same before and after glycogen depletion, since fat metabolism does a pretty good job of keeping the energy levels high. Scanning question 31. I see one-letter amino acid codes with the number indicating position, some of which also appear in the answer choices. Looking at the question more closely, it's asking why A100, F252, and E376 mutations result in MCAD dysfunction. It also mentions that mutations in R164 and M165 have no effect on MCAT deficiency, but I'm going to ignore that detail since none of the answer choices include those two amino acids. To me, this question comes out of left field. I don't remember the passage talking about any of these amino acids, nor did I get a good sense of the detailed information about the catalytic mechanism, but it's worth exploring. I'm going to head to paragraph 2, where MCAD was discussed. As I suspected, there's nothing here that relates to the amino acids, and I know from my earlier analysis the rest of the passage doesn't help, so this might be more of a pseudo-discrete question. Before I take a closer look at the answers, I think it's worth exploring if there's a pattern in the amino acids, because I know from experience that MCAT questions like to test amino acids based on their side chain properties. I know that A is alanine, which has a methyl group for its side chain. F is phenylalanine and has a phenyl group, and E is glutamate and has a carboxylate side chain. Since any of these point mutations will cause MCAD on its own, the reason for dysfunction has to be true of all three of these amino acids, so I'm going to focus on any similarities. While I can see that both alanine and phenylalanine have side chains that are hydrophobic, glutamate is certainly not. At this point, I'm not really sure what else to recall, so I'm going to go through the answer choices and eliminate the ones that don't work with the available information or aren't scientifically supported. A says that the three amino acids participate in hydrogen bonding, but that is only true of glutamate, not alanine or phenylalanine. In order to participate in a hydrogen bond, the side chain must have at least a dipole of some sort to be a hydrogen bond acceptor, or a nitrogen-hydrogen or oxygen-hydrogen bond to be a hydrogen bond donor. In any case, it doesn't work, so I can eliminate A. B says that the three amino acids are involved in disulfide bond formation, but only cysteines have the capacity to make disulfide bonds. Eliminate B. C states that all three dictate the folding of the enzyme's active site. I suppose this is plausible. This is a pretty broad and general explanation, and if it's true that all three are involved in folding, then mutating these amino acids would indeed break the enzyme. However, it's not something I really anticipated or know as a fact, so I'm going to take a look at D as well. D is also fairly general, saying that all three are needed for proper subunit interaction and catalytic properties. Initially, this was puzzling to me since C and D seem to be saying the same thing. However, D has the detail about subunit interaction, and that makes me want to look for something in the passage. Subunits means that the protein is composed of more than one polypeptide chain. So, is there any passage evidence for MCAT having more than one polypeptide chain? I'm going to take a look back into paragraph 2 again about MCAT, and here I spot the clue I need. MCAT is a monomer. That means there's only one polypeptide chain for MCAT, and there cannot be any interactions between multiple polypeptide chains, which is what that answer choice is referring to with subunits. Since MCAT is a monomer, there's no subunit interactions, which means that D is incorrect. That's all I need to confidently pick answer choice C as the correct answer. The thing that immediately stands out to me when I first look at the question 32 is the structures in the answer choices. They all have this really long hydrocarbon tail, so the question must be looking for a specific type of fat. There is an interesting pattern in the structures, though. I see that there are polar head groups for every molecule, and that the head groups differ for each of the answer choices. And then there's also the different fatty tails, length, saturation, and number. This tells me that there are multiple opportunities to target what would work for the correct answer. Reading into the question stem, it mentions something about mass spec, but that seems rather extraneous to me. Rephrased, the question is really asking what fat would be higher in patients with MCAD deficiency, so probably something that's being built up as a result of MCAD not working. I'm going to start my investigation in paragraph 2, where MCAD function was first described. Taking a closer look at paragraph 2, this detail about the reaction catalyzed catches my attention. I'm told that it functions in beta oxidation of fatty acids 6 to 12 carbons long. I know that beta oxidation takes fatty acids and chops off 2 carbon bits and adds them to CoA to create acetyl-CoA. So if I have non-functional MCAD, I'll get a buildup of fatty acids that are 6 to 12 carbons long. I know from my pattern analysis of the answers earlier that this doesn't let me make any conclusions about the polar head groups, but I can figure out if any of the answers have a hydrocarbon tail that is 6 to 12 carbons in length. It turns out that there's only one of these answer choices that has a hydrocarbon chain that is between 6 and 12 carbons, answer choice A, which has 8 carbons in the tail. All of the other answer choices have hydrocarbon tails that are much longer than 12. Taking a look at question 33, I see two big things: the fairly complex set of reactions in the answer choices, and the molecule of palmitic acid in the question stem. Another thing that stands out is that every single reaction has a C16 compound in the reactant side, which I'm guessing is the molecular formula of palmitic acid, but vary in the number and distribution of molecules like NAD+, FAD, water, and CoA. The question is asking about the reaction for breaking down palmitic acid for a person with MCAD deficiency. The advantage with chemical reactions as answer choices is that there are usually multiple ways to target the correct answer, often by utilizing the patterns in the answer choices. The first thing I'm going to do is remind myself about the exact reaction that MCAD catalyzes from the passage, which was found in paragraph 2. The reaction is described in word form in the first 2 sentences. I learned that it takes fatty acids that are 6 to 12 carbons long and produces acetyl-CoA, NADH, and FADH2. I know that the carbons from the initial fatty acid will be found as acetyl groups in the acetyl-CoA. It means that there's some other enzyme that's going through the process of breaking down longer fatty acids like C16 palmitic acid until it gets to the C12 length. Then MCAD takes over to process it down to just acetyl-CoA. This means that when I have normal functioning MCAD, I can take that C16 and break it all the way down to acetyl-CoA and the electron carriers. Without MCAD, I'm stuck at the C12 level, so I should see C12 on the product side. I'm going to see if that's enough to get me to my answer. And it turns out that's all I needed to select answer choice A. The C12 fatty acid in the product side is the key difference. B might be tempting if I stuck with the description in the passage about MCAT, where it works on C6 to C12 fatty acids, but we would never get to the C6 fatty acid level without functional MCAT. Both C and D are missing the medium chain fatty acid on the product side. Additionally, D has the wrong order for the electron carriers. The reduced form of NADH and FADH2 should be on the product side, not the reactant side.